Have you ever wondered how scientists can detect viruses like COVID-19 or measure gene expression from just a tiny amount of sample? The answer lies in a technique called quantitative polymerase chain reaction, or qPCR for short. Unlike conventional PCR, which only shows whether DNA is present after amplification, qPCR measures how much DNA is produced in real time. One of the ways in which this is enabled is by combining amplification and fluorescence detection in the same sealed tube, allowing scientists to quantify genetic material accurately, sensitively, and without post-PCR processing. One of the key innovations that makes this possible is the TaqMan probe, a molecular design that turns DNA replication into a measurable flash of light. So, how does a TaqMan probe actually work? It's a short single-stranded DNA oligonucleotide labeled with a fluorescent reporter dye at the 5' end and a quencher at the 3' end. When the probe is intact, the quencher absorbs the reporter's energy through a process called fluorescence resonance energy transfer, or FRET. This keeps the reaction dark, meaning no fluorescence, until the probe is cleaved during amplification. During qPCR, the reaction cycles through three main stages: denaturation, annealing, and extension. First, the double-stranded DNA separates. Then, primers bind to each end of the target region while the TaqMan probe hybridizes between them. As the Taq DNA polymerase extends from the primer in the 5' to 3' direction, its exonuclease activity encounters the probe and cleaves it. This releases the reporter from the quencher, producing a burst of fluorescence that signals a successful amplification event. With each cycle, the fluorescence increases proportionally to the amount of DNA produced. The point at which fluorescence rises above background, the cycle threshold, or CT value, reflects the starting quantity of the target sequence. This mechanism underlies many of today's molecular diagnostic tests. TaqMan-based assays are used worldwide to detect infectious diseases like SARS-CoV-2, monitor viral load in HIV and hepatitis B, and measure gene expression in clinical and research settings. By turning every amplification event into a flash of light, TaqMan probes illuminate the invisible world of nucleic acids, one fluorescent signal at a time.